Assalamu alaikum. Welcome to all the participants. We have a very important session. I request the host to please share the poster on the screen. It is about Antibiotic Awareness Week and microbial resistance. I request Dr. Sadia to raise her hand so that we can make her co-host. Assalamu alaikum. I would request Dr. Sadia to please start. Assalamu alaikum, Dr. Sadia. You can begin your lecture. Are you able to hear me?
तो वो है ना मैडम आपका ब्लूटूथ है तो ब्लूटूथ ऑन करना पड़ेगा। Hello, Assalamu alaikum. Sorry, class, for the delay.
Walaikum assalam, Dr. Sadia. We have doctors today from
Okay.
across India. Okay, we have almost 36 participants. Okay, Dr. Sadia, so you can begin the lecture. We can see the screen and you are
Okay.
audible also.
Okay,
Mashallah.
madam. Okay, madam. Assalamu alaikum, dear friends. I'm Dr. Sadia. I'm working as a microbiologist at Shadan Institute of Medical Sciences. At the outset, I want to thank management of SALAMAH Hospital for giving me the opportunity to talk about antimicrobial resistance and stewardship. Uh, this week in November, this month is denoted as the Antimicrobial Awareness Week, which was started from 2015 by WHO, and every year since 2015, November 16th and November 24th, this is considered as the Antibiotic Awareness Week. So today, today's topic is about the various mechanisms for the antimicrobial resistance and what are the preventive measures and the stewardship program which has to be implemented in every hospital. The objectives of my talk are the definitions of antimicrobials. These topics I will deal with in the coming slides. Mechanism of action of the various antimicrobials, the problems that arise with the use of antimicrobials, then the problem statement that is pertaining to the scenarios in world and various countries, including India, the factors which are responsible for the causation or emergence of the antibiotic resistance, consequences of the antibiotic resistance, origin and mechanisms of resistance, the detection method, that is how do we detect the antibiotic resistance and how do we control. Then finally is the antimicrobial stewardship program. Actually, this should be dealt as a different topic, but I have just added, I'll just tell a gist of what the antimicrobial stewardship program is. Now to start with, what is an antimicrobial? It is a substance which is produced naturally by one microorganism and is effective against various other groups of microorganisms. That is the bacteria, virus, fungi, and parasites. These antimicrobials, they are usually produced naturally by one set of organism. For example, penicillium. Penicillium is the first antibiotic which was discovered by Alexander Fleming in the year 1929, and it came into use from 1940s. This was discovered from the fungus Penicillium notatum. So it is this antimicrobial is produced by from one microorganism, but it is active against other microorganisms. It could be a bacteria, it could be a virus, it could be a fungi or a parasite. These antimicrobials, based on their spectrum of action, they can be bactericidal or bacteriostatic. Bactericidal is when it kills the bacterium. Bacteriostatic, which when it only inhibits the growth of the microorganism. Then next is the antibiotic. What is an antibiotic? This is also a substance which is produced by a microorganism, or it can be synthesized from a semi-synthetic substances, okay, which at low concentrations can kill or inhibit the growth of another microorganism. So next terminology is minimum inhibitory concentration and minimum bactericidal concentration. So these points are necessary. You need to know the terminology and the definitions of these, because in the coming slides, I will be using MIC, MDCs, so that it should not be something foreign to you. So if you know the definition, that will be easier for you to understand my slides. What is minimum inhibitory concentration? It is the lowest concentration of a antimicrobial agent that prevents visible growth of the microorganism. That is, lowest dose of जो antibacterial जो है, which will prevent the growth of a microorganism. That is the minimum inhibitory concentration. Minimum inhibitory concentration plays a major role when prescribing antibiotics in a hospital setting. MIC is considered as the gold standard for determining the susceptibility of an organism to antimicrobials and is therefore used to judge the performance of all methods of susceptibility testing. Better the MICs, that is lower the MICs, it will be more susceptible. The drug is more susceptible, and hence a drug which with a low MIC should be the choice for the treatment of empirical treatment of the disease. Now coming to the MBC, which is the minimum bactericidal concentration. This is the lowest concentration of a antimicrobial agent required to achieve bactericidal killing. Both are one and the same and both are interdependent. The lowest broth dilution of the antimicrobial that prevents the growth of the organism on the agar plate. When the MIC is low, only then the optimal MBC can be procured. Okay. Now, coming to the classification of the antibacterial agents. Antibacterial agents, there are different types of classification based on their spectrum of action. They are classified as narrow spectrum, broad spectrum, and extended spectrum based on the type of the action, whether they are bactericidal or bacteriostatic, which we have already discussed in the previous slide. Bactericidal is that antimicrobial which kills, whereas the bacteriostatic which only inhibits the growth of the microorganism but does not kill. Now coming to the spectrum. The antimicrobials are classified as narrow spectrum, broad spectrum, and extended spectrum. Narrow spectrum of antibiotics are those which are active only against a set of microorganisms. For example, vancomycin. Then you have the cefoxitin, then linezolid, levofloxacin, teicoplanin. So these antibiotics are active only against gram-positive microorganisms like Staphylococcus aureus, Enterococcus. Next, you have the gram-negative antibiotics. Example, aztreonam, polymyxin B. Then you have the cephalosporins, piperacillin, then so on and so forth, which are active only against the gram-negative bacteria. Now coming to the extended spectrum or the broad spectrum, the antibiotic covers both gram-positive and gram-negative in a broad spectrum range. When it comes to the extended spectrum, the antimicrobial is able to cover both gram-positive, gram-negative, and also anaerobes. Now, this is the third classification, which is based on the mechanism of action. They are antibiotics which inhibit the cell wall synthesis of a bacteria, which include the beta-lactam group of antibiotics, which are the penicillins, cephalosporins, monobactams, carbapenems. In addition to that, we also have glycopeptides like vancomycin and also other antibiotics like cycloserine. So what do these antibiotics do? They disrupt the cell wall synthesis. Cell wall synthesis होने से inhibit करते हैं ये antibiotics. So because of that, they are able to kill the bacteria. And these cell wall synthesis inhibitors are usually bactericidal antibiotics. So next group of antibiotics are those which inhibit the protein synthesis. So protein synthesis is brought about by the ribosomes which are present in the bacterial cell. So there are two subsets for the ribosomes, which is the 30S, which is the smaller unit, and 50S, which is the larger unit. There are antibiotics which inhibit the 30S subunit of the ribosomes. What are they? They are tetracyclines, then streptomycin, pectinomycins, and then aminoglycosides like gentamicin and amikacin, then kanamycin and nitrophilans. So what do these do? They inhibit the protein synthesis. Then the other group of antibiotics like macrolides, which include the erythromycin and chloramphenicol, clindamycin, and lincomycin, they inhibit the 50S unit of the ribosomes. For the protein synthesis, this 30 and 50S ribosome, they have to unite and only then they can participate in the synthesis of proteins. So, which is inhibited by these antibiotics, and hence the protein synthesis in the bacteria is disrupted or it is inhibited, and then there is cell death of the bacteria. Now, the third group of antibiotics are those which act on the cytoplasmic membrane structure. That is, they cause the depolarization of the cytoplasmic membrane, which include the polymyxins and the daptomycin. Because of the disruption of the cytoplasmic membrane, there is cell lysis and there is apoptotic death of the bacterial cell. Now, the fourth group of antimicrobials include those which act on the enzymes like DNA gyrase and the RNA transferase. What do these do? RNA dependent RNA transferase. What do these enzymes do? They help in the transcription and translation of the genetic material. So, these enzymes, when inhibited by the antibiotics like actinomycin, nalidixic acid, ciprofloxacin, and novobiocin, there is no generation or there is no synthesis of the genetic material, which is the core object for every living cell. So these and enzymes, they are inhibited by these antibiotics, therefore, and thereby the genetic material is not produced and it leads to the death of the bacterial cell. So these are the various classes of antibiotics according to their mechanism of action. Now, coming to the antimicrobial use and misuse. Today, see, we have encountered so many number of uh, bugs, which are called as the superbugs, which are resistant to a number of antibiotics. So, what could be the reason for the emergence of these antibiotic resistant strains? The most important cause is use and overuse or misuse of the available antibiotics. So, what happens when the available antibiotics are used or they are misused or overused? It leads to toxicity, that is, nephrotoxicity or ototoxicity. They might even cross the blood brain barrier, um, affecting the fetus. Teratogenic effect is there. Then it leads to cyto hypersensitivity reactions, then emergence of antimicrobial resistance, superinfections, nutritional deficiencies, masking and action, and increase the cost of healthcare. You know that when we start giving appropriate dosage, appropriate type of antibiotic, what does it lead to? It will increase the emergence of the bacterial strains which are resistant. With the emergence of the resistant bacterial strains, the patient has to stay for long in the hospital. It increases the hospital stay. So, which in turn, which increases the cost of the healthcare and it adds to the economic crunch of the country. Now, coming to the antibiotic resistance. So, what is antibiotic resistance? For the past 10 slides, I've been talking about antibiotic resistance, antibiotic resistance. So, what it is actually? It is the microorganism's resistance to an antimicrobial drug that was once able to treat. That is, an antimicrobial drug which was able to kill or inhibit the growth of a particular microorganism. But now it is unable to do the same action. That character is or that property of the microorganism is called as antibiotic resistance. It refers to the unresponsiveness of a microorganism. Previously, it was responsive, so it was killed. Now it has become unresponsive. So, this phenomenon of tolerance is called as antibiotic resistance. Now, this resistance is a property which is of the microbe, but not it is not pertaining to the antimicrobial agent or a person, or even the organism which is infected by another microbe. There is, for example, a bacteriophage. All types of microorganisms can develop resistance irrespective. It can be a bacteria, it can be a virus, it can be a parasite, it can be a fungi. Antibiotic resistance is a major subset of antimicrobial resistance. See, antimicrobial substance includes those which we give against bacterial infections, fungal infections, parasitic infections. But antibiotic resistance is a part of this antimicrobial resistance that applies specifically to bacteria that become resistant to various classes of antibiotics. Bacteria that are considered extensively drug resistant or totally drug resistant. These are called as superbugs. What do you mean by extensively drug resistant? That is, when a microorganism becomes resistant to more than three groups of available antimicrobials, that is called as extensive drug resistance. And when the microorganism becomes resistant to the possible available antimicrobials, it becomes totally drug resistant. Such such microorganisms are called as superbugs. For example, there was emergence of such superbug, which is the New Delhi metallo-beta-lactamase, which was resistant to every possible antibiotic. It was even resistant to the carbapenem. So there are very few, or maybe there are no antibiotic regimens which can be prescribed against new metallo-beta lactamase, except for cholesterol. Now, the uncontrolled microbial resistance is acknowledged as a major health crisis. WHO in 2005, it has declared antimicrobial resistance as a global threat to the health of the people, leading to the global economic shocks, and also it affects the development of the country. Irrational use of antibiotics can adversely affect the patient. What do you mean by irrational use? And what is rational use? That is, you have to use antibiotics only when indicated. When the infection is not because of a bacteria, but if it's because of a virus, you don't need an antibacterial. Then you have to treat the infection, not the colonization, nor the contaminants. So that comes under the irrational. Then use of inappropriate antibiotics. Certain infections are self-limited. So in such conditions, you don't require an antibiotic. So even in that case, when you start prescribing antibiotic, that becomes an irrational use. So this irrational use of antibiotics can lead to emergence of antibiotic resistance and increase the cost of healthcare. Antibiotic resistance, it occurs naturally, but misuse of antibiotics in humans and animals is accelerating the process. Coming to the problem statement. So I'm going to talk about the scenario about the antibiotic resistance in various countries of the world, including the developed countries like United States and UK. According to a survey which was done by CDC in 2019, more than 2.8 million antibiotic resistant infections they occur in United States every year, and around 35,000 people die out of this antibiotic resistant infections in USA, and accounting to 7 lakh deaths annually all over the world. So now the WHO has told that if this death rate it increases or it goes on this pace, by 2050, the antimicrobial resistance death toll could be up of 4.15 million in Africa, almost all the similar numbers in other, even in in developed countries. But by 2050, Asia would top the list of deaths due to antimicrobial resistance, which could see 4.73 million people dying of this antimicrobial resistance over the same period. Now I have explained about the numbers. What could be the number of deaths if it is not properly controlled? In addition to that, we are facing a problem because of the emergence of these organisms which are responsible for the number of deaths which I have mentioned below. Before, what are they? Staphylococcus aureus. So most important is the gram-positive coccus, which is the Staphylococcus aureus, MRSA, methicillin-resistant Staphylococcus aureus, which is the most common cause of antimicrobial resistance. Previously, it was observed in only 20%. Now it has increased to about 80%. That is 80% of the total Staphylococcus aureus isolates are methicillin resistant. Next organism is the Enterococcus species, which leads to vancomycin-resistant Enterococcus, which are mostly seen in hospital care settings and most common cause for bloodstream infections. Next, the Escherichia coli, which is a gram-negative bacilli. In these gram-negative bacilli, they are production of certain enzymes called ESBLs. So this gram-negative bacilli like Escherichia coli, Klebsiella, Pseudomonas, they produce ESBLs, MBLs, and AMC-beta lactamases, which are most important trouble-causing organisms. So next organism is a Klebsiella pneumoniae, which is resistant to third generation cephalosporins, which occurs in 60% of the Klebsiella pneumoniae isolates. And newer antibiotic resistance is seen to carbapenems, where the percentage of antibiotic resistant isolates are about 50%. The next organism is a Pseudomonas aeruginosa and Acinetobacter baumannii, which are also resistant to carbapenems. Drug resistance is also encountered in Mycobacterium tuberculosis. 10% of the total Mycobacterium tuberculosis isolates usually encounter this drug resistance. And most commonly, it is towards the isoniazid or streptomycin, which are the first line of drugs used for the treatment of the TB. Now, next is the antimicrobial resistance threats. I have told you the general ones which have emerged long back. Now, present scenario in 2019, a survey was conducted by the CDC and it has given this list of the organisms which could be urgent threat or cause serious threats, concerning threats or watch list. That is, what are they? Carbapenem resistant Acinetobacter, Candida auris, and then Clostridium difficile and carbapenem resistant Enterobacteriaceae. So the organisms which come under urgent threats, they are now the most important cause of antibiotic resistance, leaving hardly any treatment modalities left out. Now, serious stress. The serious threats include drug-resistant Campylobacter, Candida. So these are the various organisms which have been classified by the CDC under the various categories so that the antibiotic resistance control program or the surveillance or the rational use of antimicrobials can be decided based on the type of the threat it causes. Now, this is the timeline of the various antibiotics and their resistance. The upper segment shows the discovery and the lower segment shows the resistance, that is the emergence of resistance. The first antibiotic was salvarsan, which was first discovered by Paul Ehrlich. He used salvarsan for the treatment of syphilis. Then came in the penicillin, sulfonamides, then so on and so forth. There were a discovery of various antibiotics. But after 1987, there was no discovery of any new antibiotic. There is a wide gap of 30 years for the discovery of new antibiotics. Now, coming to the emergence of the antibiotic resistance. The first antibiotic resistance was first encountered in 1941 for the penicillin. Penicillin was called as a wonder drug when it was discovered. Before the 20th century, when we were in the pre-antibiotic era, it was very difficult to treat infections. And people used to die because of the untreated infections. The mortality and the morbidity rates were very high. Thanks to Alexander Fleming for discovering this penicillin, it became a wonder drug. So more number of infections were being treated and people were becoming healthy. In 1945, uh, Alexander Fleming, he received the Nobel Prize for his discovery of penicillin. At that time itself, he had told if the proper use of antibiotics is if not implemented, it would definitely lead to antibiotic resistance, which we are seeing it. Now coming to the emergence of antimicrobial resistance. How do the antimicrobial resistance emerge? The bacteria which are present in our body or on the various surfaces, all are not resistant. Most of them are susceptible. But what is the reason for them to become resistant? The use of or overuse of only one class of antibiotic, it leads to selective pressure on that susceptible bacteria. So because of the selective pressure of the antimicrobial on the susceptible bacteria leads to the mutations in the genes present in the susceptible bacteria. So because of that, it will mutate into a resistant strain. Now, when an antimicrobial is added or it is used for the treatment, the susceptible bacteria, whatever which are left will get killed, but whereas the resistant bacteria will dwell and whatever resistant genes it has gained through mutations, it will spread to the other microorganisms. So this is how first it undergoes mutation because of the selective pressure caused by the antibiotic. Next, it will transfer this resistant genes to the other related or unrelated microorganisms of the same group or the different group. Now, this is what I have explained previously. So the first step is in a population of bacteria, one bacterium mutates and becomes antibiotic resistance. Now, antibiotic kills off the bacteria except for the resistant ones. Now, this resistant bacteria multiplies exponentially and then forming a population of antibiotic resistant bacteria. Next, now this antibiotic resistant bacteria, now it can transfer the mutation to the other bacteria. Now it has gained the resistant genes. Now it will spread the genes to the other bacteria, making the other bacteria also resistant to various antibiotics. Now, what are the factors which lead to the antibiotic resistance? They are classified as drug-related factors, environmental factors, prescriber-related factors, and patient-related factors. So, what are the environmental factors which lead to the emergence of the antibiotic resistance? That includes huge populations, overcrowding, then poor sanitation, increased community acquired resistance, ineffective infection control programs in various hospitals, widespread use of antibiotics in animal husbandry and agriculture, and as medicated cleansing products. So, what does this mean? The first point, huge populations and overcrowding. See, because most of the people live below the poverty line and more number of people dwell in a small place. This leads to overcrowding. Because of the overcrowding, there is every possibility for the spread of the disease causing organism and thereby even the antibiotic resistance is spread likewise. The other reason is poor sanitation. The people who dwell in our community should follow proper sanitation, proper personal hygiene. See, if they keep themselves clean and neat, so they can keep the infections at a bay. So once the infection is away from you, so there is no point of gaining antibiotic resistance organisms. Now, ineffective infection control program. A proper infection control program has to be maintained in every hospital. A effective infection control program, it is the gist and it's the mainstay of every hospital for its proper functioning. What are the various infection control programs? One is hand hygiene protocol. Second is wearing proper PPE whenever indicated. Third is proper disposal of the biomedical waste. All these infection control practices has to be taken care of and they should be implemented. In absence of these infection control programs, it might lead to the spread of resistant microorganisms and then so and so forth, the antimicrobial resistance. Widespread use of antibiotics. It's not only the human beings who use the antibiotics. Now the antimicrobials are being used in animal husbandry, that is in goat, chicken, that is fowl feed and all. They are supplemented with these antibiotics. Why do you think they supplement? According to these stakeholders, once they use these antibiotics in their feed, it would increase their yield. In order to increase the yield of their flesh, that is meat and milk, antibiotics are being used. For example, it was found around 7 years back that colistin, which is the high-end antibiotic, which was added to the feed of the fowl. So now once when we consume this poultry or the meat, organisms are present in the flesh of the fowl or the goat. So these indirectly affect our health consequences. And also in agriculture. See, most of the people now they say organic farming. You please eat, consume food or consume food items which are grown by organic farming. Normally, they are using preservatives or pesticides which are harmful to our body. Now, the second group of factors include the drug related. Everywhere in India, it is very easy to get antimicrobials. Over the counter availability of antimicrobials is there. So unless you cut down the availability of antimicrobials over the counter, till then the antibiotic resistance cannot be controlled. Then counterfeit and substandard drug causing suboptimal blood concentration. That is usage of substandard drugs will lead to suboptimal blood concentration. That is the amount of concentration, that is the therapeutic dose which is required that cannot be met if you use a substandard drug leading to suboptimal blood concentration. Now, irrational fixed dose combinations of antimicrobials. Some of the doctors or some of the physicians, they use fixed combinations, which is not acceptable. Now, next, soaring use of antibiotics. This is nothing but overuse or misuse of the antibiotics, which leads to a selective pressure on the anti against the microorganisms. Now, patient-related factors, which include poor adherence of dosage regimens, poverty, lack of sanitation concept, lack of education,
Dr.
self-medication.
Sadia, थोड़ा सा
हाँ,
उर्दू भी चाह रहे हैं सब लोग. Sorry to disturb you. They want a little bit of Urdu also. Just
okay. So patient-related factors कौन सी है? Poor adherence of dosage regimens. मतलब आपके डॉक्टर आपको एक एंटीबायोटिक एक पूरा एक कोर्स लिख के देते फॉर 5 days या 6 days. So you have to adhere to that number of days. If doctor has writing कि you have to take medicine for 5 days, 5 दिन खाना है एंटीबायोटिक, तो आपको 5 दिन खाना ही है. ऐसा नहीं कि 2 दिन के बाद चलो मैं ठीक हूँ तंदुरुस्त हो गई. आप you should not stop your antibiotics at day 2 or day 3. पूरे कंप्लीट 5 days या 6 days जितने भी दिन आपके डॉक्टर लिखे यू हैव टू टेक एंटीमाइक्रोबियल्स फॉर दैट मेनी नंबर ऑफ डेज. So this comes under poor adherence of those regimens. Moreover, now the dose what you take, जो भी हम लोग एंटीबायोटिक की खुराक हम जो ले रहे हैं, the dose should be optimal to the person who is taking it. For example, an adult. For an adult, I would prescribe. अब antibiotic लिख रही हूं मैं. It is 500 milligram of amoxicillin. It is BID. That is 2 times a day. Now, what if I eat 250 milligrams of antimicrobial once in a day? Do you think that is optimal dosage? No. So this has to be taken care of. जब आपके डॉक्टर लिख रहे हैं आप 500 मिलीग्राम एंटीबायोटिक खाना है 2 टाइम, तो you have to adhere to the dose. करेक्ट डोसेज खाना जरूरी है क्योंकि पीडियाट्रिक ग्रुप के वास्ते डोज अलग है और एडल्ट ग्रुप के वास्ते डोज अलग है. नाउ पॉवर्टी, बिकॉज ऑफ पॉवर्टी, गरीबी, बहुत सारे पेशेंट्स अफोर्ड नहीं कर पाते टू बाई दैट कॉस्टली एंटीबायोटिक्स. देन लैक ऑफ सैनिटेशन कॉन्सेप्ट. तो मोस्ट ऑफ द पीपल दे डोंट नो सैनिटेशन का मतलब क्या है. अपने आप को साफ सुथरा रखना है, हाथ धोना है, एंड खाना खाने से पहले या किसी पेशेंट को हम लोग ट्रीट कर रहे हैं, किसी पेशेंट को हम टच कर रहे हैं, तो इमीडिएटली यू टच, आफ्टर यू टच द पेशेंट you have to wash your hands. Next, lack of education. Most important thing is lack of education. The people are not educated properly. They don't know about antimicrobial क्या होती है, कहाँ पर एंटीबायोटिक लेना है, कौन से इन्फेक्शन को लेना है, कौन से इन्फेक्शन को नहीं लेना है. So वो चीज हम लोगों में कमी है. So that we should take care of. Next is self medication. वो तो बहुत कॉमन है आजकल. मेडिकल शॉप पे जाते हैं, वो लोग बोलते कि देखिए भाई मेरे को नजला है, मेरे को गले में खराश है, तो मेडिकल शॉप वाला दे देता. वो डॉक्टर थोड़ी है, उसको थोड़ी मालूम कि आपको कौन सा एंटीबायोटिक जमता और आपका हेल्थ कंडीशन क्या है, कुछ कोमॉर्बिडिटीज या कुछ डायबिटिक है या हाइपरटेंसिव है. बस थोड़े एंटीबायोटिक्स थोड़े ग्रुप ऑफ पेशेंट्स को नहीं देना चाहिए. सो दिस इज सेल्फ मेडिकेशन विच इज वेरी डेंजरस. नेक्स्ट इज मिसकंसेप्शंस. पीपल थिंक दैट चलो एक दिन खा लिया या कोई जरूरत नहीं है, चलो भाई हम लोग जाएंगे वहां पर दुआ पढ़ा लेंगे, एंटीबायोटिक क्या दवा की जरूरत नहीं है, हम लोग ठीक हो जाएंगे. दैट इज अ मिसकंसेप्शन. जहाँ दुआ है वहां पर दुआ काम आती, जहाँ दवा खाना है वहां पर दवा खाना चाहिए. Now prescriber related. This is pertaining to the doctors. Inappropriate use of available drugs. फर्स्ट ऑफ ऑल, जो भी डॉक्टर लिख रहा है, ही शुड पहले उसको करंट नॉलेज होना चाहिए जो नए एंटीबायोटिक्स कौन से है और कौन सा एंटीबायोटिक कौन से इन्फेक्शन को देते हैं. वायरल इन्फेक्शन में एंटीबायोटिक की जरूरत नहीं है, तो उसको पता होना चाहिए कि अरे ये वायरल इन्फेक्शन है तो मैं एंटीबायोटिक क्यों दूं. मोस्ट इंपॉर्टेंट, हम लोग इवन मेनी पीपल they have eaten azithromycin, they have eaten ivermectin in COVID infection. Do you think this ivermectin and azithromycin might have any role in COVID infection, which is a viral infection? So this is one example because of the lack of current knowledge and inappropriate use of the antibiotics. Now, increased empiric poly antimicrobial use. When a patient comes to a doctor, अब आपको तो नहीं मालूम उसको क्या इन्फेक्शन है, उसको कौन सा एंटीबायोटिक सूट करता या सही इन्फेक्शन है नहीं है, तो आप कैसा कंफर्म करोगे. पहले तो यू स्टार्ट विथ वन स्मॉल और बेसिक एंटीबायोटिक और general which is active against many group of organisms, like pehle I have discussed, that is the broad spectrum antibiotic or extended spectrum antibiotic. The doctor should start with the empirical poly antimicrobial use because it is not aware of which antibiotic has to be given. But this empirical treatment also should be given before, should be given after the sending the patient for the chat box. Also there are certain questions. Yeah, at the end of the session I'll try to explain your questions about susceptible. Okay, fine. Yeah. Now, so next factor is overuse of antimicrobials. Matlab, the same point, that is it goes around the bush, that is beating around the bush. जहां पर एंटीमाइक्रोबियल एंटीबायोटिक जरूरत है वहां पर जितनी खुराक चाहिए, जितने दिन के वास्ते चाहिए, उतना ही हम लोग को लेना है. उसके आगे we are not supposed to take the antimicrobial. This is called as overuse or misuse. Now, inadequate dosing, that we have already explained. I have already explained in the previous topic, that is previous factor, that is patient related. The doctor should write optimum dosage based on the age of the patient, उसके weight के ऊपर, weight के हिसाब से, age के हिसाब से, he should write the correct dosage. Okay, inadequate dose लेने से जो बैक्टीरिया अपने जिस्म में है, the infection, it will not be cured properly. Complete इलाज नहीं होता, फिर भी the bacteria is still in your body. Now it will become resistant to the antibiotics. Now coming to the consequences of antibiotic resistance. So up to I have discussed about the factors which cause antibiotic resistance. Next, what is a problem statement? कहाँ पर कितने deaths हुई हैं? कौन से organisms है which is causing antibiotic resistance? Now I will tell about the consequences. अगर antibiotic resistance को control नहीं किया गया, तो उससे क्या complications हो सकती है, क्या after effect हो सकती है. First is the lead to the therapeutic failure or relapse. इसका मतलब है जो हम लोग दवाइयां ले रहे, they could be a failure. इससे पहले in the previous slide I told you inappropriate use of antibiotic. बोले तो जहां पर सही एंटीबायोटिक सही डोज में अगर नहीं दिया गया for the correct time, it might lead to emergence of resistant strain. बोलते that bacteria which is present in your body will become resistant. So because of that, हमारे पास जो regular antibiotics है, they will not work on this microbe. Okay, so there is no complete treatment and there is relapse of the infection. Again, आपको फिर से वही infection होने के chances है, but with a organism which is more stronger than the previous one. Second is it forces the physician to use newer antibiotics because the physician is left with hardly any antibiotics for the treatment of the infection. तो अब क्या करेगा doctor? He will look to newer antibiotics. The newer antibiotics which can be costly. Also, it can cause toxicity in the body. That is, it might lead to harmful effects because of this antibiotics. Then can spread in the hospital environment. अगर infections हो ठीक से timely अगर treat नहीं किया गया, hospital में ये resistant organisms will spread एक इंसान से दूसरे इंसान, one patient to another patient, and because of that it will lead to infections which cannot be treated. तो अगर antibiotic resistance उसको कंट्रोल नहीं किया गया, तो ये जो भी रेसिस्टेंट ऑर्गेनिजम्स है बहुत ज्यादा तादाद में स्प्रेड हो जाएंगी. उस वजह से ऐसे इंफेक्शंस आएंगे जो नॉर्मल एंटीबायोटिक से ट्रीट हो ही नहीं सकते. So has to face prospectus of untreatable infections. So this is the outcome of a antibiotic resistance organism. This is drug of choice has changed over the years. That this इसका मतलब है because of the untreatable infections, तो person will be affected with infections which cannot be treated with the normal. That the drug of choice has changed. That is now the doctors have to think about नए antibiotics, newer method. But what is the use of prescribing new antibiotics? Again, that will be used, overuse हो जाएगा, misuse कर लेंगे. फिर again new antibiotic mechanism, resistance mechanism आएंगे. तो finally कोई antibiotic बचेगा ही नहीं. Now coming to the types of antibiotic resistance. Antibiotic resistance दो types की होती हैं. One is the environmentally mediated drug resistance, then microorganism mediated drug resistance. Environmentally mediated drug resistance. Any changes in the physical or chemical characteristics of the environment will lead to this environmentally mediated drug resistance. ये drug resistance जो है वो environment में कोई तब्दीलियां like physical characteristics या chemical characteristics में अगर कोई तब्दीलियां आ रही है, that will also lead to antibiotic resistance. Example is pH. Next is the presence of cations like magnesium and calcium. ये जो ions है magnesium की और calcium की, अगर ये ज्यादा तादाद है बॉडी में, उस वजह से ये जो अमाइनो ग्लाइकोसाइड एंटीबायोटिक है वो काम नहीं करता। ये कौन से ऑर्गेनिज्म में देखा गया है? सुडोमोनास एरिजिनोसा। Sorry. The second type of resistance is microorganism mediated drug resistance. That is, this is caused due to the genetically encoded trait of the microorganism. पहले तो environment related था, अगर environment में कुछ भी chemical okay, but now next is microorganisms. तो microorganism में ऐसी कौन से changes है, ऐसी कौन सी changes है जिस वजह से resistance आगे बढ़ सकता या spread हो सकता? They are intrinsic resistance and second is the acquired resistance. Intrinsic resistance मतलब क्या है? It results due to the resistance from normal genetic, structural or physiological state of microorganism. I'll tell you. The microorganism, बोलते जो भी germ है, जरासीम है, या it can be a microorganism, it can be a virus or a bacteria, because of the genetic structure जो उसमें normally है, because of that different genetic structure, it will cause intrinsic resistance. Example is gram-negative bacilli. They are intrinsically resistant to vancomycin, antibiotic, and gram-positive cocci. तो दो किस्म के organisms होती हैं, gram-positive cocci, that is gram-positive organism and gram-negative organism. जो gram-positive organism है, वो naturally वो resistant होती हैं to antibiotic like estrionam, linezolid, levofloxacin. Next is, sorry, polymyxin B, not levofloxacin, sorry, to estrionam, polymyxin B, and certain cephalosporins. But coming to the gram-negative bacilli, that is gram-negative bacilli, जो organisms हैं, they are naturally resistant. That is, this antibiotic cannot work against the gram-negative bacteria. What are they? Vancomycin, then comes the linezolid, levofloxacin, then the teicoplanin. So these antibiotics cannot be used for gram-negative bacilli. What are the mechanisms? किस वजह से ये intrinsic resistance हो सकती है? They lack the target. That is, there is no cell wall around the bacillus. Lack of the cell wall will make the organism naturally resistant to certain antimicrobials called the penicillins. Then innate efflux pumps. What do you mean by efflux pumps? Bacterial surface में there are certain pumps called as the efflux pumps. What do this efflux pump do? जो भी एंटीबायोटिक एंटर्स इनटू द बैक्टीरियल सेल, जब वो एंटीबायोटिक या एंटीवायरल जब ऑर्गेनिज्म या माइक्रो ऑर्गेनिज्म के अंदर जब आती है, तो इमीडिएटली व्हाट हैपेंस इज द इफ्लक्स पंप्स व्हिच आर प्रेजेंट इन द सेल वॉल, उसकी जो सेल वॉल में जो इफ्लक्स पंप्स है, इमीडिएटली दे विल फ्लश आउट दिस एंटीबायोटिक्स और एंटीवायरल्स टू द एक्सटीरियर। तो ये ऑर्गेनिज्म वो जो भी एंटीबायोटिक है उसको सेल में एक्यूमुलेट होने नहीं देती है, एंड इट विल पुश आउट ऑल द एंटीबायोटिक व्हिच हैज एंटर्ड इनटू द सेल. दिस इज कॉमनली सीन इन एस्करिशिया कोलाई एंड सुडोमोनास एरिजिनोसा. देन ड्रग इनएक्टिवेशन. द ड्रग व्हेन वी, अब देखिए हम लोग एंटीबायोटिक्स लेते हैं, इट विल अफेक्ट और इट विल एक्ट ऑन द microbe. But what happens is, in certain organisms like Klebsiella, it produces an enzyme. Enzyme produce करते हैं, that is a cephalosporinase enzyme. That cephalosporinase enzyme will break the antimicrobial or the antibiotic. उस वजह से the antibiotic becomes ineffective. Okay. Now, next is the acquired resistance. So, intrinsic resistance, I have told you, it is by its genetic makeup. like how we have innate immunity by our virtue of birth. We get immunity from our mother by the virtue of birth, and due to our genetic makeup, we are able to fight diseases. That is the innate immunity. Similarly, the organisms may be उसकी genetic makeup की वजह से they are naturally resistant. So that comes in intrinsic resistance. The second type of resistance is acquired, means to gain from exterior. बाहर से they get from outside. That is called as acquired resistance, acquiring, gaining from outside. Now, acquired resistance develops by mutation or acquisition of new DNA. इसका मतलब है, when does acquired antibiotic resistance occur? When there is mutation. See, I have told you in the previous slides, overuse of antibiotics will cause selective pressure on the bacteria and there is mutation. So because of the mutation, it becomes a resistant strain. So here also the same implies. Acquired resistance develops due to mutation, that is changes in the genetic material or the chromosomes. Then acquisition of new DNA, either existing gene में या तो तब्दीलियां आएंगी या तो नया gene from exterior they will gain to become resistant. Now coming to the origin of antibiotic resistance, there are two origins, genetic and the non-genetic. Genetic may I have told you, it is the mutation, changes in the DNA is called as the mutation. Now coming to the non-genetic origin. Non-genetic origin occurs in organisms like Mycobacterium, where which is intrinsically resistant. मतलब है ये organism जो है वो dormant है. It divides at a very slow pace and it keeps itself shielded in the cells. So that is the reason the host defense mechanisms do not identify this, and treating this bacteria becomes difficult, which leads to the emergence of drug-resistant mycobacteria. Now coming to the genetic mechanisms. So what are the various genetic mechanisms which can lead to antimicrobial resistance? So first is the mutations. Mutations occur in the chromosomes. Then gene transfer. Gene transfer is transferring one gene from one organism to the other organism. That is called as gene transfer. So how does this gene transfer occur and what happens because of gene transfer? So first is the by transformation, वो ये एक मेथड है, दूसरी ट्रांसडक्शन है और तीसरी कॉन्जुगेशन है. ये तीन मेथड्स है जिसके थ्रू ये जो एंटीबायोटिक रेजिस्टेंस कॉजिंग जीन्स जो है एक बैक्टीरिया से दूसरे बैक्टीरिया में ट्रांसफर होती हैं. Now what are those extra chromosomal genetic material which will help in the drug resistance? एक तो प्लास्मिड होती है, दूसरी ट्रांसपोजोन and third is the integrons. May I know who is not able to understand? Can you please tell who's not able to understand? Please raise your hand.
Ji.
Like someone has messaged that it's becoming difficult because actually this
I
is
have told them this is only for doctors because we are having the WHO awareness week of antibiotics. So some of the students of our class, no, pregnancy and child care, for them they are finding it difficult. So I explained to them this is only for doctors. तो म्यूटेशन जीन ट्रांसफर ये सब हम लोगों को ही समझ में आएगा ना, उनके लिए थोड़ा सा मुश्किल होगा. जी.
जी
How
मैम.
many more minutes, Dr. Sadia? Like you have fitness. Yeah, you want to continue it tomorrow because you said you will take it in 2 parts.
Yeah, madam, this next part about the biochemical mechanisms and antibiotic stewardship, I'll continue in the next class on Saturday.
Saturday. Okay. 2:30. Okay.
Yes,
ठीक है.
madam.
आज का आपको अभी तो थोड़ी देर लगेगी शायद complete. Right? Right. Okay.
हाँ, madam, because it's a bigger topic. That's why.
Okay. Sure. Thank you.
Okay. Okay. So now I have told about the mechanisms, that is the genetic mechanisms. एक तो which are mediated by plasmid, which is an extra chromosomal genetic element other than plasmid, and the plasmid which relates to the antibiotic resistance is called as the R plasmid, which contains two subunits, that is the resistance transfer factor and the R determinant. R determinant carries the resistance gene. Next are the transposomes, which are also called as the extra chromosomal genetic material or also called as the jumping genes. What do these transposons do? How do they transfer the genetic material? They carry or they transfer this genetic material from one chromosome to another chromosome or from chromosome to a plasmid. Now, this mechanism of antibiotic resistance, which are the biochemical reactions, which will be dealt in the next class. And after that, we will also I will talk about the various preventive measures, how to diagnose the antibiotic resistance, what are the various methods, then the antibiotic stewardship. I'm sorry, some of you are not able to understand. This is actually basically for the doctors, which madam has already explained. I'll try my level best to explain in Urdu. Madam, should I end the meeting? I'll continue in the next class.
No, we can take 1 or 2 questions, Sadia, from the chat box. ठीक है. Susceptibility जो है, एक क्वेश्चन था आफिया का, susceptibility to infection, like you are prone, यानी कि आप उतने कमजोर है, right? That is how it is. You can explain, Sadia. Yes.
In general, any person who is healthy, मतलब उसको कुछ और problems नहीं है, comorbid conditions नहीं है, diabetic नहीं है या hypertensive नहीं है, ऐसे लोग they are not prone, मतलब उनमें infection आने के chances कम होती हैं. But whereas individual जिसमें diabetes है या hypertensive है या कुछ और दूसरे medical conditions है, उन लोगों में immunity level कम होती है, जिस वजह से they become prone, मतलब they'll be more likely they'll be infected, risk ज्यादा रहता है इन लोगों में infection आने की. One such category is the pregnant woman. Even a pregnant woman is more prone to infections. Okay, which I have explained previously. There is a change, that is उसकी immune system में तब्दीलियां होती है, जिस वजह से pregnant women infections होने के risk बहुत ज्यादा हो.
Next session, like आप एक session फिर Saturday जो लेंगे Dr. Sadia, so it will be followed by a quiz also.
हाँ. Yes,
तो आप कुछ questions आपके ही lecture के questions आप देंगे, okay?
जी मैडम.
आज का lecture और Saturday का lecture दोनों का content के साथ आप एक quiz हमको देने वाले हैं, right? तो all doctors जो specially आज के lecture सुने हैं, हम आपको YouTube link भी भेज देंगे. आप again you can listen to that, so that आपको कुछ भी अगर समझ में नहीं आया, you can definitely put down your questions. Or once it is completed, we will definitely give you the PPT and the material also. Right now, we will be sharing you the link, YouTube link, where this lecture is recorded. So I'm very much thankful to Dr. Sadia Shehnaz Sahiba. She is MD microbiology. She handles the lab in SALAMAH Hospital. She is assistant professor in Shadam Institute of Medical Sciences, Hyderabad. Jazakallahu khair. Thank you to all the sisters who are present. I think Sister Mujawir is having one doubt. Yes, you can please tell what is your doubt. You can raise your hand also. We will unmute you.
Okay.
जज़ाकल्लाह जी, आप अपना क्वेश्चन चैट बॉक्स में भी लिख सकती हैं या फिर यस मुजाविर साहिबा, if you have. And with this we will end today's meeting. Jazakallahu khair. Assalamu alaikum wa rahmatullahi. Yes, sisters who have personal issues, like you can just share it directly. Okay, we will definitely try to solve it. Jazakallah. Assalamu alaikum.
Alaikum salam.